So my name's Kristy. What I'm going to talk about today is my summer experience with the DNA vaccine construction, testing, and strategic management of beef cattle internship. So I did a lot of cool things this summer. We went to the Reproductive Biology Center to collect blood from anaplasma positive and anaplasma negative cows and their calves. This blood was used for research on an ongoing anaplasma vaccine. Then my major part of my summer was focused on my TET-ON system construction, which is my research project. And I performed a lot of procedures to get this going, and those are listed up there. Also, since I've done so well during the summer in Dr. Cooper's lab, I've been offered a student worker job next semester and to work on a myostatin vaccine, and I am currently writing that undergrad research proposal for that. And these are just some pictures. Obviously, the cows are at the RBC, and then those other two are in lab. Also, obviously, the workforce development was a large part of this internship program, and the competencies I chose are listed up there. It has made me a lot more aware of myself and the skills I have and lack, and I will definitely pursue working on those in the future. As far as career and educational plans, I will most definitely go to graduate school, whether that is at LSU or out of state, and my end career goal will be to be a reproductive specialist. Okay. So what my project is to construct a TET-ON system for the progression of DNA vaccine production. So currently in lab we have several DNA vaccines being researched, and those include brucella, anaplasma, salmonella, and more recently myostatin. So the TET-ON system will work in unison with another project called the ZER project to create a stable vaccine that won't require refrigeration while keeping its immunogenicity. The TET-ON system works by controlling a gene of interest, which in our case is REC-A, by inducing the system in the presence of a tetracycline derivative such as doxycycline. Two major components in this system are the TET response element and the reverse TET repressor. These work to bind to doxycycline, for example, in its presence rather than the TET response element allowing transcription to occur, thus inducing the system. And that first graphic shows how that works. Moving to my materials and methods, each component was found in literature review and individually purchased. Each of these were integrated into the plasmid, which was for us GP704, and they were altered as needed depending on what we needed it to do. And following these alterations and integrations, there's a transfection of our S5327 strain of E. coli and a collection of that newly constructed DNA. And since this research is still in progress, we don't have any definite results yet, but as soon as the construction is finished, the product will be sent for sequencing, and that will confirm our orientation of our vector. And after that, we will be running an antibiotic plating system on this to see what cell line that we have is the most stable, since we have antibiotic resistant genes, and technically they should grow on these antibiotic plates. And following that, we will ligate the ZER project into it, and what this does will take out the antibiotic resistant genes and replication of origins from the vaccine. And this is very important because this is what makes the vaccine stable and won't require refrigeration. This is more geared towards developing countries since they don't have easy access to electricity, let alone refrigeration. and hopefully this will deter epidemics of preventable diseases and improve the welfare of animals and people around the world.